Prior to the beginning of the 20th century, infectious diseases accounted for high morbidity and mortality worldwide. By 1900, some of the leading causes of disease were pneumonia, tuberculosis, diphtheria, and intestinal disorders like diarrhea and enteritis. Together, these diseases caused one-third of all deaths. Of these deaths, 40% were among children under the age of 5. The average life expectancy at birth during this time was only in the mid-40s, in part due to infectious disease. If you were a soldier in the military during the Civil War or any of the wars leading up to World War II, you were more likely to die of an infection than an actual injury, regardless of the care of a physician. For soldiers and civilians alike, the dirtier a wound was, the more likely an infection would result, and once an infection started, it was unlikely that the victim would survive. The discovery of penicillin in 1928 by Sir Alexander Fleming was a game changer. By 1942, two scientists, Ernst Chain and Howard Florey, had figured out how to purify and stabilize the drug, and it was tried in humans as a treatment. Its use in the military saved countless lives, and by 1945 the drug was widely available to civilians as well. This work ushered in the discovery of many new antibiotics, and the period between 1950 and 1970 was named the Golden Era of the discovery of novel antibiotics. After that, the approach to the discovery of new drugs was modification of existing antibiotics. The antibiotic era revolutionized the treatment of infectious disease worldwide. Here in the U.S., the leading causes of death changed from communicable diseases to non-communicable diseases like cardiovascular disease, cancer, and stroke. The average life expectancy rose to about 78.8 years. Infectious diseases have become a problem of the elderly, cancer patients, transplant patients, surgical patients, and patients on immunosuppressive drugs, and other at-risk groups in developed countries like ours. Let us not get too comfortable, though. Infectious diseases are still among the top 10 cause of death in the U.S., and with the introduction of COVID-19, it's on the rise again. The objectives for this video are to define selective toxicity, distinguish between broad-spectrum and narrow-spectrum antibiotics, distinguish between bactericidal and bacteriostatic, and to name 5 cellular targets for antimicrobial drugs. Let's start with a little vocabulary. If I say the word chemotherapy, what comes to mind? Probably cancer treatment, right? Well, in the 1920s, chemotherapy was a term that was used to mean the use of chemicals to treat infectious disease. Nowadays, chemotherapy broadly means the use of drugs to treat a disease, whether it's a cancer drug, anti-cancer drug, or whether it's antibiotics or other antimicrobials for infection. So it's a broad term that means the use of drugs to treat a disease. Antimicrobial drugs are any drugs that interfere with the growth of microbes within a host. These can be naturally derived drugs, but they can also be synthetic drugs that a scientist or a chemist has made in the laboratory and found effective. Antibiotics, for this class, we're going to use a narrow definition. So I know out in the public and maybe even in your workplace you use antibiotics really to mean any kind of antimicrobial, but for this class, antibiotics specifically refer to substances produced by microbes. Very often these microbes are molds or fungi. They can also be bacteria. But in small amounts, these substances inhibit the growth of another microbe. So for example, some bacteria produce antibiotics to inhibit other similar types of bacteria from growing. This improves their ability to survive in a highly competitive environment. Remember back in chapter 14, we talked about microbial antagonism? Antibiotics are a chemical that some microbes use to antagonize or inhibit the growth of other microbes. A definition that you definitely need to remember for your exam is selective toxicity. Your optimal antimicrobial drug is going to be one that is harmful to a specific microbe, but it doesn't harm the host that has the microbe inside. This is what we mean by selectively toxic. So these selectively toxic drugs are going to target structures that are part of the microbe but are not shared with the host. So I hope at this point you're thinking about all those times I asked you about the cell wall and all those times I asked you about ergosterol and chitin. Those would be great targets for selectively toxic drugs because they are only in the microbe. They are not shared by most hosts. When we talk about antimicrobials, there's a number of terms that we use to describe their activity. One such term is broad spectrum. Broad spectrum drugs affect a range of microorganisms. They don't just treat gram-positive microbes, but they treat gram-negatives as well, for example. Some examples of drugs that are broad spectrum antimicrobial drugs include augmentin, tetracycline, and anipidem. If you think about the microorganisms in your body, they are a mix of gram-positive and gram-negative, and there are yeasts too. Some of these microbes will have inherent resistance to antibiotics, just because of their genetics or their interactions with other microbes. I've drawn a few microorganisms on this slide. I've indicated their inherent resistance using plus signs. So where there's a 0, that means no resistance to antimicrobials. Where there's 1, there's a little resistance. Where there's 2, there's more resistance. And when there's 3, there's maximal resistance. I've even included a pathogen here for you. Now, when you take antibiotics or antimicrobials, the most susceptible microbes are the ones that are killed first, followed by those that have only slight resistance. And within a few days to a week or so, the more resistant microbes start to die. By the end of treatment, hopefully, the pathogen will have also died. However, with fewer neighbors around to compete with, the resistant microbes, whether they're the pathogen or not, they can grow unimpeded. Further, they may pass their resistance on to other innocent microbes by conjugation or transformation. As a result of broad-spectrum antibiotic usage, antibiotic resistance may be favored. Now, as you are aware, antibiotic resistance is a major problem in healthcare today. The CDC maintains surveillance programs for dozens of antibiotic-resistant microbes, some of which are multi-drug resistant, meaning that they're resistant to more than one antibiotic or antimicrobial. And a few are actually pan-resistant, meaning that they are resistant to all known antibiotics. Narrow-spectrum antibiotics are those that are of limited effectiveness because they only affect one type of microorganism. For example, they may only be effective against gram-positives like penicillin G, or they're effective against mycobacterium like ethambutol. These drugs are given when a doctor has done a test and knows what specific microbe is causing an infection. This type of treatment is called a definitive therapy. The risks associated with this type of treatment are lower because there's less damage to the normal flora and the treatment is more targeted. It's the difference between using a wrecking ball and using a sledgehammer to take down a wall. Both will do the job, but one is more precise and less destructive than the other. Here's a table of antimicrobial drugs. Which of these listed drugs has the broadest spectrum of activity? Tetracycline, of course, because it's effective against gram-negative bacteria, gram-positive bacteria, and it's even effective against intracellular parasites like chlamydias and rickettsias. How about a narrow-spectrum drug? Can you find that? Right, there's a lot of choices here. You've got penicillin G, there's isoniazid, there's ketoconazole, there's praziquantel, acyclovir, all sorts of drugs you could have chosen here. They would be narrow spectrum because they only affect a small number of cells. Before we leave the topic of broad spectrum and narrow spectrum antibiotics and antimicrobials, I want to revisit superinfection one last time. Remember, superinfection is a risk of broad spectrum antibiotics and antimicrobials. In this phenomenon, the normal flora is so damaged by a drug that microbes like yeasts or resistant bacteria which are not affected by the drug grow out of control because the competition for survival has been reduced. An example of this is when some people take broad-spectrum drugs to treat a bacterial infection. They sometimes become susceptible to yeast infections or C. diff infections. There are fewer bacteria around to outcompete these pathogens, so they grow out of control and may cause some debilitating effects. Two more terms that we need to be familiar with are bactericidal and bacteriostatic. Bactericidal drugs kill bacteria directly. For example, penicillin inhibits cell wall synthesis. Without a strong cell wall, bacteria burst due to osmotic lysis. The activity of the antibiotic directly leads to the death of bacteria. Bacteriostatic drugs, on the other hand, inhibit the growth of bacteria so that the host's defenses, particularly phagocytic cells of the immune system, can kill the bacterium. One misconception is that bacteriostatic drugs are weaker than bactericidal ones. Something to keep in mind is that the line between bacteriostatic and bactericidal is fuzzy. It really boils down to dose. Some drugs are too toxic to use at their bactericidal concentration, so they have to be used at lower doses and are thus bacteriostatic. An example of this would be tetracycline. Other drugs are called bacteriostatic, but they're actually bactericidal. It's just that they're only bactericidal at higher concentrations than are needed to inhibit growth. Thus, we call them bacteriostatic. The last thing that we'll talk about in this introductory video is the 5 ways that antimicrobial drugs inhibit the growth of microbes. We'll talk about bacteria specifically in this and following lectures, but the same concept is applicable to other living cells. What's that concept? The concept is that antimicrobial drugs should target structures that are in microbes but are not shared by the host. This goes back to selective toxicity from the beginning of this lecture, which said that a selectively toxic drug is one that will harm the microbe but not the host. And the way that you do that is by targeting structures that are in the microbe but are not in the host. So let's take a look at some of them. The first one is the cell wall. So one target for your antimicrobial drugs is cell wall synthesis. Human cells do not have cell walls, so that these drugs are effective and generally not toxic. They're only effective against actively growing microbes, and in the case of bacteria, these drugs have to inhibit the proper synthesis of peptidoglycan. With a weakened cell wall, bacterial cells become susceptible to lysis. Your second target is protein synthesis. So some of your antimicrobial drugs target protein synthesis in bacteria versus protein synthesis in humans or other eukaryotes. Remember, proteins are required for life, and there are no organisms that can survive without proteins. Ribosomes make protein. Can you recall any difference between protein synthesis in bacteria and protein synthesis in eukaryotes like us? Back in chapter 4, we said that bacteria have a 70S ribosome and eukaryotes have an 80S ribosome. This implies that for your prokaryotes and your eukaryotes, there is a different ribosome structure. And this means that there are antibiotics that are naturally available that are targeted more towards prokaryotes than eukaryotes. Because remember, the whole reason, well, I might not have mentioned it, but the whole reason we have antibiotics is because fungi and bacteria have been in an arms race for eons, trying to outcompete each other for survival, and they've produced antibiotics to try to kill other microbes that are nearby, making more food and more resources available to support their own survival. We just kind of figured out what they were doing and figured out ways to kind of extract these proteins and molecules that they had already developed. So in any case, over hundreds of thousands, even billions of years, these organisms have devised proteins and other molecules that are specifically designed to target the 70S ribosome, not the 80S. So that means that these drugs that target protein synthesis are somewhat selectively toxic. Now, I will tell you that there are some classes of antibiotics that can be toxic to eukaryotic cells because they're able to penetrate the plasma membrane. And remember that our mitochondria have ribosomes that are similar to the 70S ribosomes. A third target for your antimicrobial drugs is the plasma membrane. Some peptide antibiotics, like polymyxin B, can change the permeability of the plasma membrane. So it can, for example, maybe bind lipids in the plasma membrane and disrupt them. This leads to leakage of the cytoplasm for your microbial cells, and that can lead to lysis. Your next target is nucleic acid replication and transcription. Many antimicrobial drugs interrupt the synthesis of DNA and RNA. Unfortunately, these drugs are of limited usefulness because in some cases they can affect DNA synthesis and RNA synthesis in eukaryotic cells. But there are some classes of drugs that target proteins that are unique to your bacteria or nucleic acids that are unique to fungi, for example. Your fifth target is essential metabolites. So some antimicrobial drugs competitively inhibit the production of essential metabolites that microbes need for producing their proteins, their DNAs, and other molecules. Antibiotics competitively inhibit synthesis of these molecules by mimicking these molecules, and they can prevent chemical reactions from happening. When these chemical reactions are prevented, then a compound that's needed for another reaction isn't available, and depending upon what that compound is, this can lead to death. Later in these lectures, we'll talk about an antimicrobial called PABA, which mimics a metabolite necessary for making amino acids and nucleic acids in bacteria. The drug mimics PABA and prevents the chemical reactions from happening. Before I wrap this video up, let's do a little check-in to see what you remember. Here's your first question. Selective toxicity. Is it number 1, describes antimicrobials that are toxic to host kidneys? Number 2, describes antimicrobials that kill only bacteria? Number 3, describes antimicrobials that harm microorganisms but do not harm hosts? Or number 4, describes antimicrobials that are toxic only under specific circumstances? What do you think? Did you say describes antimicrobials that harm microorganisms but do not harm hosts? If you did, that's right. Don't forget to pause the video while you think of your answer and then press play when you want to check and see if you're right. Here's another one. Some microbes in the normal flora overgrow or outcompete other microbes due to antibiotic resistance or damage caused by broad-spectrum antibiotics. Is that number 1, unfortunate? Number 2, a primary infection? Number 3, biofilm formation? Or number 4, superinfection? Superinfection. That's right. Let's try one last one. Which of the following is not a common target for antimicrobial drugs? Would that be cell wall synthesis, cellular carbohydrates, protein synthesis, or plasma membrane? Cellular carbohydrates. That's correct. So in this video, we defined selective toxicity. We said it is the property where a drug is harmful to a microorganism, but not to the host. And remember, the way that we get to selective toxicity is by targeting structures that are in the microorganism, but not shared with the host. We also distinguish between broad-spectrum and narrow-spectrum antibiotics. Remember, your broad-spectrum antibiotics are those that are going to target more than one group of microorganisms, and that comes with its own risks. Narrow-spectrum antibiotics are less risky, and they target specific groups of microorganisms. Broad-spectrum doctors give when they're not sure what's causing an infection, but the infection is severe enough that it needs a treatment. Narrow-spectrum, the antibiotic is usually, the doctor has already done a test somehow, and they know what's causing the infection. One thing that I will say is that sometimes a doctor will give a prescription for a broad-spectrum antibiotic while they're waiting for the test to come back on what's causing the infection, and then they may switch to a narrow-spectrum. The other thing that we did was we talked about the difference between bactericidal and bacteriostatic. Remember, bactericidal, the drug is going to directly kill the microorganism. Bacteriostatic means that the drug inhibits the growth of the microorganism so that the host's own immune system can kill the bacterium. And one thing I will tell you is that although we've talked about bactericidal and bacteriostatic, these suffixes cidal and static can be used in other ways like virucidal or fungicidal or fungistatic. So just keep that in mind. Lastly, we talked about 5 cellular targets for antimicrobial drugs. We said that inhibiting the cell wall, inhibiting protein translation via the ribosomes, inhibiting nucleic acid synthesis, inhibiting the plasma membrane, and inhibiting the synthesis of essential metabolites are all ways that antimicrobials try to eliminate or inhibit the growth of microorganisms in hosts. In the next video, we're going to start drilling down and looking at specific types of antimicrobial drugs and antibiotics. I look forward to seeing you there.